This is the last of our cardiovascular system chapters. We've seen the blood, we've been through the heart, and now we're going to end with peripheral circulation, which is just blood flow through the body. You'll see some functions of circulation listed there, including transport, and transport includes just about anything that would be contained within the blood, such as hormones, components of the immune system, enzymes, nutrients, wastes, gases like carbon dioxide and oxygen. So that kind of takes us into number 2 a little bit as well. We also have regulation of blood pressure and directing blood flow. Directing blood flow in the sense of keeping the blood flowing in the right direction, but also preferentially sending blood to certain parts of the body in some instances. So it's going to be helpful for us to just examine the different kinds of blood vessels that are part of the circulatory system. There are 3 types of vessels: arteries, capillaries, and veins. And I'm listing them in the order in which we would encounter them as the blood leaves the heart, so we can kind of create maybe a picture or a story, follow some kind of chronology as we are tracing blood flow through the body. The individual vessel types do differ in terms of their size and composition. So if we start with the arteries, the list under the arteries now follows a size gradient. So we are beginning with the largest arteries, which are the elastic or conducting arteries. The next largest are the muscular or distributing arteries, and then we have the smallest arteries called the arterioles. And for the first 2, the elastic and the muscular arteries, the name really describes a lot about the composition of the wall of the artery. So in the largest vessels, there is a lot of elastic tissue. Now, the elastic arteries, being the largest ones, would include things like the aorta. That's the largest artery in the body. And these arteries are going to be the ones that are closest to the heart. And so it helps for them to be a little bit elastic or stretchy because they are the arteries that are bearing the brunt of the blood pressure that is being generated by the contraction of the heart. So the blood that is immediately pumped out of the heart, you're going to have the highest pressure right in that area. And then the pressure kind of goes down a little bit as you move farther away from the heart. Which then takes us to the second type of artery, the muscular. So these arteries are a little bit smaller in diameter, although they're still a pretty good size. And as the name suggests, they have quite a bit of muscle, a smooth muscle lining that is incorporated into those arteries. And so those muscular arteries rely a little bit less on the force of the blood coming straight from the heart because they are a little farther away, but the smooth muscle is going to need to contract in order to kind of maintain the blood flow. And then, like I said, the arterioles are the smallest. Now, after the blood leaves kind of the artery side of things, we reach the capillaries. And the capillaries are the smallest blood vessels of all. And it is in the capillaries where we have exchange. So that's kind of like trading things off, mainly exchange of carbon dioxide and oxygen. So the oxygen would leave the blood, the carbon dioxide would come into the blood, and the oxygen would then be delivered to the tissues. Carbon dioxide would go back to the lungs to be expelled. So, like I said, these are the smallest vessels. And then on the way back to the heart, we encounter the veins. So I think we had mentioned before in the previous chapter, arteries take blood away from the heart and veins take blood back to the heart. And kind of that turning point, that turnaround point is the capillary. So the veins, in comparison to arteries, they have thinner walls. And so the individual layers that comprise the walls are not as represented. They're a little bit less elastic, and they're a little bit less muscular than arteries. And they do have structures inside called valves, which help uh, get the blood, keep the blood moving in the direction of the heart, and keep any backflow from taking place. So as we go through the different kinds of veins, I'm actually switching the order of them compared to how I did the arteries. With the arteries, we looked at the largest to smallest because that's what we're going to encounter as we leave the heart. On the way back, what we're going to see is after the blood leaves the capillaries and goes now onto the vein or into the vein side, we're going to see blood flow through the smallest veins first, and then the veins will get progressively larger as we approach the heart. So the venules are the smallest veins, and then we actually have a group that are called small veins, but they're a little bit larger than venules. And I should clarify, when I talk about the size of these things, I'm referring to the diameter of these vessels. And then the largest veins are going to be your medium or large veins. They don't really have special names like the arteries, but some of the largest veins would be the brachiocephalic vein. We also have the vena cava superior, the superior and the inferior vena cava that drain directly into the heart. So those are the largest veins in the body, and then at that point we're back in the heart. So I want to look a little bit more closely at the capillaries and how that exchange takes place. This is a capillary network. Since the capillaries are so small, where they do kind of congregate and cluster, you're going to see a sort of a whole grouping of them. And once again, we see the red and the blue, which means exactly what it meant in the other chapters. The red vessels are carrying oxygenated blood, and the blue vessels are carrying deoxygenated blood. If you look closely at the picture, the largest vessels really that are shown in this picture are the arterioles and the venules. Okay, and so those are the smallest of the actual arteries and veins, and you can see what comes after that or before those are the capillaries. So you're having the arrows there are indicating the direction of blood flow. Blood's coming through the arteriole and then possibly making its way through this capillary network, going through the red capillaries first. Those are the arterial capillaries. And then you'll see kind of a changeover to the blue color, because presumably what is happening in the transition or at the location of the transition from red to blue is that the oxygen is leaving those capillaries and making its way into the cells, which you can maybe imagine kind of in the surrounding area. And then that blood becomes deoxygenated. So then as we continue our route through the network, the blood all of a sudden changes to blue as the oxygen is no longer present. That blue blood also has carbon dioxide in it that the cells have dropped off, but keep in mind, really, it's not the carbon dioxide that is explaining the color of the blood. It's the oxygen levels, either being there or not being there. And so we continue through the blue capillaries until we get to the venule, and the venules are going to join up with the larger and larger veins on the way back to the heart. One of the things also you might notice, especially in relation to the red capillaries, the arterial capillaries, is there are little sphincters. They look like little white bands that are wrapped around some of those capillaries. And those sphincters are there to kind of shunt blood in different directions. Not all those capillaries that you see in that network may be open for blood flow. This is one of the ways that the body is able to direct blood flow to different parts and also regulate excuse me, the rate or the amount of blood that is reaching a particular area. And so it's possible that some of those sphincters, as they kind of tighten around those capillaries, would prevent blood flow going through certain routes Okay, so the blood would have to take different avenues. It could still get through. There'd still be an exchange taking place, but the blood doesn't have to fill every single capillary in our body at all times, and it doesn't. Some of the vessels like that are shut down at any given time. So to kind of summarize the exchange in terms of how it works, okay, and once again, the exchange is just takes place in the capillaries and where we have movement of materials both in and out. And the two major ways that things move in and out of capillaries are through diffusion, which means that there is they move from an area where they are more concentrated to an area where they are less concentrated. Okay, and depending upon where we are in the body, that may be into the capillary or it may be out of the capillary. So things like glucose, amino acids, oxygen, hydrogen ions, wastes, carbon dioxide, those are relatively small molecules, and they can fairly easily diffuse through most capillaries. They can go right across the cell membrane of the cells that line the capillary walls. They can go in between the individual cells that line the capillary walls, and those are the intercellular spaces. Or in some capillaries, there are actually little gaps that occur between the cells that line the wall, and those are called fenestrations, which are literally little windows. The other type of transport that we see at work here is mediated transport, and that occurs only in some areas such as the brain. And mediated transport is a little bit more of a selective type of transport. It utilizes proteins that are embedded in the cell membrane, and like I mentioned, it allows the cells to be a little bit more selective in what they allow through and maybe when. So the way that the blood moves through these capillary beds, as you saw in the previous picture, when the blood is flowing, starting from the heart, going through the various levels of arteries and such, there's going to be fluid movement out of the capillaries at the arterial end, and then on the venous end, in the vein capillaries, most of that fluid will return back to the bloodstream. So in other words, things get pushed out of the arterial capillaries, but then they very often, most of that material will re-enter the venous capillaries. Whatever does not re-enter circulation is picked up by the lymphatic system, which we will be covering in a later chapter. And then eventually, though, the lymphatic system does indirectly join up with the circulatory system, and we get back to some of our big veins, and that material joins back up with the regular blood flow. When we look at the movement of a vessel, really, whether it be an artery or a vein, what you're seeing here are the dynamics of circulation and how the movement of the blood actually happens in relation to the vessel. There are two types of blood movement. We have laminar and turbulent flow. Laminar flow is a very streamlined, quiet type of flow, which you'll see pictured in letter A. And what you'll see here is the red arrows represent the blood that is filling that portion of the vessel. You'll notice that all the arrows are parallel to one another, so it's a very streamlined type of blood flow. Everybody's going in the same direction. And the concentric layers represent the speed at which the blood flows, okay? And so that large arrow right in the middle is indicating that the blood is flowing fastest right through the center of the vessel. And as we kind of radiate out from there, the arrows are getting shorter and shorter because the blood flow is slowest then in the layer that is right against the vessel wall. Okay, and you kind of think of it this way. Let's say you're walking down a really crowded hallway where people are more or less shoulder to shoulder. The people that are kind of in the middle of the hallway, as long as they keep moving, should be able to move pretty freely and pretty smoothly through. Whereas the people that are up against the wall, they may actually be physically pushed against the wall. Their shoulders may be scraping the wall, and so there's a little bit of what we would call in this instance resistance to their movement. And that's what the blood layers that are close to that vessel wall are experiencing. They're experiencing a little bit of resistance. And so there's definitely an asymmetry in terms of the pace and the ease at which the blood can get through that vessel. On the other side, in letter B, you'll see another vessel. Looks a little bit different because the kind of that part on the right-hand side is a little bit narrower. This vessel is constricted. Okay, it means that it's a little bit smaller. It's not uniform all the way through. Now the thing about vessel size and blood flow is, just think about how much space this blood has to move through. So where you have a wider vessel, you're going to have faster blood flow. Where you have a constricted or smaller vessel, blood flow is going to slow down. Okay, again, that constriction is going to impede the movement of the blood to some degree. And so what happens when you have a situation, especially where the vessel is wide open in one area, and right at the point where it starts to constrict, you're going to see more of a turbulent flow. And turbulent flow, as you can tell from the arrows, it's kind of a crazier, more haphazard type of flow. It's not smooth. There's a lot of think about when an airplane experiences turbulence. There's a lot of bumping and bouncing around of that blood, almost kind of sloshing in different directions as it's trying to make its way through that vessel. Turbulent flow happens, like in this case where there's a constriction, when there is maybe an obstacle, something physically in the way that's blocking blood flow or at least slowing it down, or sometimes when the blood starts flowing so fast above a critical velocity, it starts to kind of lose control and we get turbulent blood flow as well. So we'll kind of get into the implications of this in a little bit here. So speaking of blood flow through the vessel and the ability of the vessel to constrict or open up wider, not only does that impact the flow of the blood getting through, but it also impacts blood pressure. And blood pressure is just the force of the blood against the wall of the vessel. It's measured in a unit called millimeters of mercury. And I'm going to kind of relate this to something with which you might be familiar. When we measure blood pressure, and I know we actually did a lab when we were, back before break on blood pressure, and you guys used the digital blood pressure meters, which, just kind of gave you the numbers. But I want to take you through kind of the theory behind the measurement of blood pressure and kind of the way that they used to do it. Maybe they still do, but the more analog way of doing it. The measurement of blood pressure is based upon listening for sounds, they're called Korotkov sounds, produced by the turbulent flow in blood vessels. And I failed to mention before, but the laminar flow, I think I said, was a quiet type of flow. When blood flow is turbulent, it makes a lot of noise. Again, kind of like a splashing or a sloshing against the vessel. So you can hear turbulent flow under certain conditions. And so the measurement of blood pressure, not using the digital meters that we did, is accomplished by an instrument called a sphygmomanometer. And so that would be, you put the cuff on the arm like you guys did, but instead of hooking that up to the digital meter, you would have a stethoscope, and you would use the stethoscope to listen for these sounds. So the theory behind this is when the cuff is put on the arm and you pump up the cuff and it kind of squeezes the arm, it's collapsing the brachial artery in the upper arm. or just above the elbow, really. And that's in order to cut off blood flow. So if there's no blood flow, you're not going to hear any kind of sound of the blood moving through vessels. Now what happens then is that pressure is slowly alleviated. So in other words, you slowly kind of let the air out of the cuff, and the cuff, the pressure of the cuff, gets smaller, and you're slowly opening up that brachial artery. Okay, when that brachial artery is open enough to allow at least some blood flow through, okay, it's not going to be all the way open, but it'll be kind of like the picture in the previous slide. That brachial artery will be partially constricted. And what that'll do is it will allow some blood flow through, but that blood flow will be turbulent. Okay, and so the person who's listening through the stethoscope, at that point that they start hearing that turbulent blood flow, that is the first sound, and that is what we call the systolic pressure. Systolic pressure is the pressure of the blood during the contraction of the heart ventricles. Okay, so then the pressure continues to drop. Okay, so you keep letting the air out of the cuff, and the brachial artery in the meantime is getting larger and larger. Okay, so as soon as it's partially open enough to create turbulent flow, that's the first sound that's heard. As the brachial artery opens up wider, then that turbulent flow becomes laminar flow. Okay, because you no longer have a constriction, you have a nice wide open artery, and you have now that quiet blood flow. So the next thing the person is going to be listening for is actually an absence of that sound. Okay, so on the onset of the turbulent flow, the sound itself, that's when they record the systolic pressure. That would be the one number, the top number. And when that turbulent flow becomes laminar and the sound disappears, that is the point at which they record the diastolic pressure, which would be the bottom number. Okay, so as you may be aware, blood pressure is reported as systolic over diastolic. So for example, on average, if you have a blood pressure of 120 over 80, the 120 is the systolic pressure, and the 80 is the diastolic pressure. Diastolic pressure, and we talked about diastole in the previous chapter, that refers to relaxation of the heart. So the diastolic pressure is the blood pressure when the ventricles of the heart are relaxing. Now, blood pressure does vary in different parts of the body at any given time. Okay, so when you look at just the whole the map of the circulatory system, there are so many vessels, and it covers you've got vessels down in the lower parts of your legs, and then you have vessels that are closer to the heart. There's no way that the pressure is going to be exactly the same in every single part of the body at the same time. So when we talk about blood pressure, really what we're measuring is what's called the mean arterial pressure, which is kind of like an average or an overall pressure to represent what's going on in total. Mean arterial pressure is about 70 millimeters of mercury at birth. From adolescence to middle age, it goes up to closer to about 100, and in healthy individuals, around 110. Again, these are averages. And the mean arterial pressure is dependent upon a few things. One is the cardiac output, and that's the volume of blood that's pumped in one cardiac cycle. So the cardiac cycle is what we went over in our heart chapter, which is essentially a heartbeat. And so how much blood is pumped out of the heart in a heartbeat is known as the cardiac output. It's also dependent upon heart rate, which is measured in beats per minute. That'd be like taking your pulse. And it's dependent upon peripheral resistance. And resistance is something that opposes blood flow. So as resistance goes up, blood flow goes down. A constricted vessel would be a good example of a source of resistance. Okay, it either slows down the blood flow or in some cases may cut it off entirely. Now, blood pressure is, something that is we need to monitor all the time, constantly, and our nervous system has a hand in doing that. We have hormones as well that help regulate blood pressure, make sure it doesn't go too high or too low. And then there are also some some reflexes, some autonomic reflexes that all of these things work together to try to maintain homeostasis with respect to blood pressure. Again, make sure that it's within normal limits. So we addressed blood flow before, but I want to come back to that more as just not so much in relation to individual types of vessels. But so blood flow is, when we talk about blood flow, we're most interested in the rate of blood flow, how fast it's going. Okay, so the volume of blood per unit time. And blood flow is directly related to pressure differences, and it's inversely related to resistance. So these are some of the things that I had kind of mentioned before. So blood, as we saw from our heart chapter, flows from an area of high pressure to low pressure. Okay, so if you remember how the cardiac cycle went in the contraction and the relaxation of the ventricles, changed the volume of the ventricle, and it therefore changed the pressure. So it was, the heart is in a sense creating these different pressure gradients or pressure differences, which then facilitate the direction of the blood flow. And so this continues on even outside of the heart into the vessels themselves. So one of the things that keeps blood flowing through the circulatory system is these pressure gradients. Okay, so we mentioned before that pressure is the highest in the vessels that are closest to the heart, and that pressure sort of decreases as we get a little farther away from the heart because you're not as close to the pump. And so even when the blood has been, is quite a distance from the heart itself, you still have this gradient, okay? And so that's going to keep the blood moving from that high pressure area to the lower pressure area. And so the P1 and P2 represent pressure at, let's say, imagine a little portion of a blood vessel, okay? And you mark one area of the blood vessel and you call that P1 to represent the pressure at that particular point in the vessel. Then you go downstream a little bit and you mark another point, call it P2, which would be the pressure at point number 2, some distance away. The difference between P1 and P2 is the pressure gradient or the pressure difference. And what we're saying about blood flow here is if there is a greater difference in pressure between those 2 points, then the blood is going to move more quickly between those 2 points. It's kind of like, you can think of a gradient as like a hill. A hill that has a steep gradient versus a hill that has a smaller gradient. So the hill with a steep gradient is going to be a steeper hill. A hill with a smaller gradient would be a little bit flatter. Okay, and if you put a ball at the top of the hill, it's going to roll faster down the steeper hill. Okay, the blood is going to move faster down a steeper gradient. The inverse relationship with resistance we mentioned before, where resistance, as the term itself really suggests, it's resisting blood flow. So anything that resists blood flow, whether it be a constricted vessel or a blockage in a blood vessel, those are all things that will slow down blood flow, or at the very least, or will in some cases may stop it altogether. So the inverse relationship is that as resistance, for example, goes up, blood flow goes down. So they have the opposite, they move in the opposite direction. As resistance goes down, blood flow goes up. Now, one of the things that is probably has the most impact on resistance is the radius or the size of the vessel. I guess we could refer to the diameter as well. But that's probably going to have the most impact on a regular basis to blood flow, is just the vessels changing size. Now, one of the ways that we can help control blood flow and really to sort of maximize the effectiveness of distributing the blood, and the whole point in blood flow is to feed our cells, to get that oxygen, to get the nutrients to the cells, and to be able to take away waste products. And so it's very important that we know about the needs of the different tissues, and that's what this control of blood flow is all about. Blood flow is generally proportional to the metabolic needs of the body. And there are just some areas in the body that use more energy than others, certain types of tissues, certain organs. So metabolic needs, that what I mean by that is how much oxygen they use, how much glucose they use, basically how much energy they burn, okay, and how quickly they need energy. In parentheses there, you'll see some highly metabolic areas: the brain, kidneys, liver, skeletal muscles. Okay, not that our other body parts don't need energy, but these areas use proportionately more energy compared to others. And so, for example, they need more glucose to make more energy. Therefore, blood flow is going to be greater to these areas than it would be to a less metabolically active type of tissue or organ. Okay, so the more you need, the more you get, is kind of what it equates to. How the body knows kind of, where to send the blood then has to do with the nutrient requirements. Okay, so if there is an area of the body that is using oxygen like crazy and consuming glucose, then those nutrients are going to disappear very quickly from the blood. Okay, and those nutrient levels and gas levels are monitored. They're monitored eventually by our brain. So the brain kind of knows what all the tissues are getting. And so if you have, for example, the skeletal muscles, those are real big energy eaters. So if the muscles are demanding a lot of oxygen and demanding a lot of glucose and consuming those so that they are disappearing from the blood, well, they're going to see an increased blood flow. Now, at the same time that these tissues are using oxygen and glucose, they are processing those materials inside of the cells, and they give off waste products, which include carbon dioxide, and also sometimes things like lactic acid, but carbon dioxide is going to be the big one. And so as the oxygen and glucose disappear from the blood, those waste products are going to move into the blood and start accumulating. So the disappearance of the nutrients, as well as along with the increase in the levels of the metabolic wastes, that's an indication that these tissues are consuming these nutrients, okay, that these tissues need what the blood is supplying in a big way. And so the levels of those things, as they're monitored in the blood, impact can, let's say, can cause an increase or decrease in blood flow. Okay, and the way that this works is, so for example, if you're seeing high metabolic rates, if you're seeing high levels of carbon dioxide and lactic acid in the blood, which are waste products, then what's going to happen within those vessels that are feeding those tissues is vasodilation. Okay, and this is what I had been describing before. I don't think I ever put a name on it. Vasodilation is the dilation of the vessel, or the opening up of the vessel. Okay, so the vessel actually gets wider, the smooth muscle lining relaxes, and it opens the vessel up wider, increases its diameter. Therefore, increases blood flow. On the other hand, if you have high nutrient levels in an area, okay, nutrients now, so oxygen, lots of glucose, don't seem to be disappearing from the blood, that means that those tissues are probably not using the oxygen and glucose that quickly. Okay, and so we're not needing to send any additional blood flow or increase blood flow until they can use up those nutrients. And so that would lead to something called vasoconstriction, which is the opposite, where the vessel actually decreases in size and blood flow, let's just say, slows down a little bit, okay, or is maintained at least at a steady rate. One of the things, of course, that would go along with those high nutrient levels are low levels of waste products, because if the cells are not metabolizing, if they're not using glucose and oxygen, that means they're also not giving off much carbon dioxide or lactic acid. Mentioned there are a number of regulators in the body, including hormones and our brain, nervous system, and for example, the sympathetic nervous system is a side of the nervous system that can cause vasodilation in skeletal muscle and also cardiac muscle. If you remember, that's the side of the nervous system that is the makes us more alert, increases breathing, increases blood flow, increases blood pressure, and so those are the parts of the body that are going to receive that increased blood flow and kind of pump us up in a little bit for whatever needs to be done. Here you can see what vasodilation and vasoconstriction look like. You'll see that top vessel kind of coming down there, and wrapped around those vessels are the sphincters. We saw them back in the capillary network before. And when a vessel is dilated, like you see there on the left, you'll notice that the sphincter is relaxed. So that allows the vessel to stay pretty wide open, and you've got free blood flow going through there. Vasoconstriction, on the other hand, on the right, if you look at that top vessel, that sphincter has really tightened itself around the vessel. And so you're going to see in that case either reduced blood flow or possibly even cutting off blood flow entirely. But this is something that is that happens very frequently throughout the circulatory system, both for the purposes of regulating and directing blood flow, as well as regulating blood pressure, because kind of like in the heart, where the ventricles contract and relax and pressure changes in the ventricles, similar things happen with the vessels. So when the vessels get bigger or smaller, that impacts blood pressure as well. Okay, that does it for this chapter, and we'll be back with lymphatic and immune system next.